Hi, I'm Sietse and I'm standing in front of the Agrico Research Center. Agrico is one of the biggest Dutch potato companies, with locations in more than 80 countries around the world. Agrico played a big role in helping us develop our iGEM project. Let's head inside. According to the Food and Agriculture Organization of the United Nations, over $750 billion worth of food is lost every year before it can be consumed. To put that into perspective, that is enough to solve world hunger 3 times over. These numbers are largely due to pests and pathogens, nematodes being one of them. We are Team iGEM Groningen and we are here to put a stop to it. The potato is one of the biggest contributors to the Dutch agricultural sector. It occupies 1/4 of the country's farmlands and receives half of all Dutch agricultural subsidies. In 2017, the Netherlands exported $100 billion worth of agricultural goods, and potatoes contributed $1 billion to this number. This clearly shows how important this crop is for the Netherlands. Parasitic nematodes are the biggest threat to this industry. Among these nematodes, the biggest troublemaker for Dutch farmers is Globodera pallida. G. pallida is native to the Andes and is now present in 55 countries, predominantly in temperate regions. In Europe alone, this nematode causes crop losses of $460 million a year.
Hi, my name is Fenny, and now I will walk you through the life cycle of Globodera pallida. It involves multiple stages and begins with the eggs hatching in the presence of root secretions called exudates. When the eggs hatch, the nematodes enter the juvenile stage and move towards the root exudates. The nematodes penetrate the root tissue of the potato plant and form feeding sites inside the root that are necessary for their survival. When they enter the adult stage, the nematodes consume the nutrients and the water that the potato needs to grow and develop. The females, after fertilization, develop into new cysts, giving rise to 200 to 500 new nematode eggs. In this form, they can survive in the soil for up to 20 years, waiting for the favorable conditions and presence of a potato plant as a new host. There are several methods being used nowadays to fight off the G. pallida in the potato field. However, no method so far has been proven to be fully effective and sustainable. The use of pesticides is a common practice, but it's often detrimental to soil biodiversity. It is therefore slowly being phased out by governmental institutions around the world. Sometimes, farmers apply drastic measures, like flooding the whole field. Although it is effective, this method is very costly for the farmer and is not only limited to G. pallida. Crop rotation, the shifting of the crop grown every year by the farmer, is popular but not fully protective, and not every farmer is capable of doing this. Classical breeding methods can be an effective method, but it takes too long to develop because of the adapting G. pallida population. Genetically modified crops are developed faster, but both consumers and governmental institutions don't really consider this to be a safe solution. Considering all of these issues, we decided to develop a different solution, root patch. While developing root patch, we needed it to hold 4 key values. Firstly, since farmers need to apply it on a large scale, root patch should be easily applicable. Second, we aimed at developing a GM solution, so we had to ensure its safety and control to avoid any spread of GMOs in the environment. Third, root patch should only affect the harmful nematode, keeping the beneficial organisms in the ecosystem intact. And last but not least, since G. pallida is not the only parasitic nematode in the world, we aim to develop a general solution that could be applied to other parasitic nematodes in the soil. We were brainstorming about how to solve the nematode problem when we got an idea. What caught our attention was the work of Jonathan Dalzell and his colleagues, who explored the effects of neuropeptide-like proteins on the chemotactic behavior of nematodes. Neuropeptide-like proteins, or simply NLPs, can be found in many species of nematodes as they are involved in the regulation of their neuronal behavior. NLPs are different from the classical neurotransmitters in humans because they can modulate the activity of a whole neuronal network, rather than just facilitating single neuron-to-neuron communication. This is also why they are believed to be very significant for the nematode's functioning. NLPs also regulate the neuronal networks for sensing the environment, causing the nematodes to be attracted by the root exudate. The group of Dalzell attempted to investigate how increasing the levels of the NLP in G. pallida would affect their chemoattraction towards the exudates. They investigated this with a simple experiment. After putting the nematodes in contact with different NLPs for 24 hours, thereby changing their internal NLP levels, they were placed in the middle of a petri dish. This petri dish was divided into 3 parts.